Hello everyone, let's go start discussing about CBSE board class 12th PYQ. The chapter is molecular basis of inheritance. So let's start some conceptual question. So first question is, how many base pairs would a DNA segment of length 1.36 mm have? So in this question, length of DNA is given and asking found out how many base pairs. So first of all, I will convert it length of DNA into meter. So here the length of DNA is given 1.36 mm. For converting into meter, multiplied by 10 to power minus 3. Now we know that distance between two base pair is 0.34 into 10 to power minus 9 meter. Now we apply the formula of length of DNA segment. Number of base pair into distance between two base pair. Here length of DNA segment is given. We can write 1.36 into 10 power minus 3 equals number of base pairs which is found out into 0.34 into 10 power minus 9. Now number of base pairs after solving, the result is 4 into 10 power 6 base pair. So this is very simple questions. Now going to second question is, in an experiment DNA is treated with a compound which tends to place itself among the stocks of nitrogenous base pairs, as a result of which the distance between two consecutive base increases from 0.3 nanometer to 0. Nanometer. This is given. The length of DNA increases. Notice. Then calculate the length of DNA double helix which has 2 into 10 power 9 base pair in the presence of saturating amount of this compound. So in these questions can be solved by the same formula, like use above the question. In this question, the base pair given and length of DNA also is given. So we can apply the formula and resulted 2 into 10 power 9 into 0.44 nanometer. After solving, you can obtain the result. Now the third question is, calculate the length of DNA of bacteriophage lambda that has 48502 base pairs. In these questions, the base pair is given and asking find out length of DNA. So same questions guys, and same formula will apply. Distance between two consecutive base pairs, we know that 0.34 into 10 power minus 9 meter, and the length of DNA is bacteriophage lambda 48502 into 0.34 into 10 power minus 9. After multiplying, the result is 16.499 into 10 power 6 meter. Very simple. Next, question number 4. Why are proteins either positively or negatively charged? So, dear students, if the proteins are rich in basic amino acids, they are positively charged, and if the protein are rich in acidic amino acids, they are negatively charged. Next questions number 5. Mention the carbon positions to which the nitrogen base and the phosphate molecule are respectively linked in the nucleotide given here. So here in this diagram the nucleotide is given. You see that clearly. So the answer is nitrogenous base is linked to first carbon number. Phosphate is linked to the fifth carbon number. Very simple. You see here. Sugar is connected linked to base at carbon number first, and phosphate group linked to at carbon number 5. Very simple. Now I am coming to the question number 6. What are A and B in the nucleotide with purine represented below? You see here. Very simple questions. Here is the box B asking what is this is a nitrogen base. Very simple. And here A, this is a blank box. This is the phosphate group. So very simple. And you see here answer is A is phosphate group and B is purine, adenine or guanine, maybe. Okay. Okay, now next question number 7. Name the negatively charged and positively charged components of nucleosome. This is previous question of CBSE Delhi 2015. So very simple. In nucleosome, the negatively charged component is DNA and positively charged component is stone octamer.